Hi everybody, Fritz here. In today's video, we'll be going over your hydrolysis lab. In your hydrolysis lab, you'll be given 3 plates to represent starch for your starch, a milk agar for your casein plate, and a tributylin plate for your lipid. The reason why we're looking at each of these molecules is because these are the molecules that represent the 3 major energy resources that bacteria use to produce ATP. Starch for your sugars, casein for your proteins, and lipids representative of your fats. Now keep in mind that although each plate has a primary ingredient that separates it from the others, there is some basic nutrients in there that will allow bacteria to grow. So unlike other labs where growth on the plate indicates a positive result, growth on the plates in this lab does not necessarily mean a positive result. And we'll touch on what a positive result is in just a second. But this lab on the first day that you have your plates is relatively simple. All that you'll be doing is a simple linear streaking 2 different bacteria on each of the plates on opposite ends of the plates. And make sure to really do this on opposite ends of the plate. That way, there's no cross-contamination between the 2 bacteria. And carefully label the plates as well. That way you know which bacteria is which, because things could get moved around in the incubator. When you come back for observation, you will notice that your lipid plate out of all 3 is the easiest to tell a positive result. A positive result is indicated by a clearing or a halo around the bacteria of interest. Secondly, your casein plate. The casein plate is a little bit more difficult than the lipid plate in that the halo may not be so obvious. But a good secondary indication of a positive result is if there's some clearing or haziness around this bacteria. That is your positive result. So if you can't clearly see the halo or clearing, look at both bacteria and determine which one is cloudy or hazy, and that is indicative of a positive result. But just confirm this with your lab professor. Lastly, starch. Starch is the hardest of all 3 to tell a positive or a negative result with just the naked eye. So we need to use iodine. Now iodine does not act as a reagent or reactant in the chemical process at all. It's just a visual indicator to help us identify the positive and negative result. So a good idea here is to place a blank sheet of paper underneath this plate before adding your iodine. That way you have a contrasting background to help you identify the positive bacteria. Now that you have your positive and negative results, what does all of this mean? The purpose of this lab is to determine which of these bacteria has the enzyme of interest. In your starch plate, we are looking for the presence of amylase. With the help of water, amylase breaks down the complex structure of starch into individual components of glucose. And if you think of starch as a chain of sugar molecules that are all attached to each other, what amylase does is it breaks the bond between the individual sugar molecules to give you your glucose molecule, which is very similar to what happens on your casein plate. Your casein plate is looking for the presence of casease. This is the enzyme that breaks down the long chain of protein to individual amino acid components. And as you can see in this reaction, casease also needs the help of water to break down the protein to individual amino acid constituents. But don't be fooled here. This protein on this side of the equation is not the same protein as on this side of the equation. This protein over here is less one amino acid because casease broke it off, very similarly to how amylase breaks down starch to glucose. Lastly, we have your lipid plate, which looks for the presence of lipase. Unlike amylase and casease, lipase does not need the presence of water to break down lipids into individual glycerol and fatty acid components. And I'm sure that you're already familiar with the typical lipid molecule, which has a glycerol head and 3 fatty acid tails. What lipase does is it breaks the bonds between the glycerol head and each of the fatty acid tails to give you the respective components. And this is significant because each of these molecules can go through an energy producing process. Take glycerol for instance. Glycerol can go through glycolysis to produce pyruvate. This process on its own creates ATP, but pyruvate can further be used in other energy processes to create more ATP. And then each fatty acid tail can react with coenzyme A to become acetyl-CoA and go through other energy processes such as aerobic respiration, to create more energy, such as the electron transport chain. And this is why in a single cycle, lipids produce so much more energy than proteins and sugar, because in one cycle, you get 4 energy producing molecules compared to the process that gives you a single amino acid or a single glucose molecule in your starch and casein plate. So a fair question to ask is, which one of these enzymes in a single cycle produces the most amount of energy for the bacteria? You could also be asked, in general, what do all of these enzymes have in common? What do they do in general? They do not produce any energy for the cell. Rather, they take a complex structure and break it down to a simple structure, a monostructure. That monostructure is what is then used in further energy processes to create ATP for the bacteria. But all these enzymes do not create energy. It just takes a complex structure and breaks it down to a simple structure. And here are some additional questions that you could be asked on your lab practical. Now feel free to answer all those questions down in the comment section below. If you found this video helpful, please leave a like and subscribe, and please check out the description for additional study materials on this lab. If you have any additional questions for me, please leave it down in the comment section below, and I'll see all of you in the next one.